Welcome to the third part of this lecture on structural DNA nanotechnology. My colleague George Church said the problem with your field is that it looks like you're having too much fun. And the reality is learning how to build with these structures is fun, but we also firmly believe that the power behind it also has to do with what kinds of applications might be possible. And in particular, we're very interested in applications in molecular biophysics and future therapeutics. So I'm going to share with you some of the research directions in my laboratory trying to find useful applications for these DNA nanostructures. So the first application is we're able to create a tool that allows the NMR structure determination of an alpha helical membrane protein. This is work that was done in collaboration primarily with James Chu's group at Harvard Medical School. So the story starts again with Ned Seaman, the person who started the field of structural DNA nanotechnology. You might recall from the first part that we had this vision of flying fish, of a host guest crystal, and that maybe this would make structural biology much easier if we had access to these host guest crystals. Ned Seaman's group made an important landmark discovery along the road to this eventual goal. In 2009, they reported this nice paper where they rationally designed a unit cell. They called it the tensegrity triangle that basically has three crisscrossing double helices that define three different axes in three-dimensional space. And if they program them with sticky ends to self-assemble, then they could actually make a macro-scale crystal that has dimensions of about 0.2 millimeters per side and diffracted X-rays down to 4 angstrom resolution. So this is an important first step. A next step will be to improve the resolution of the crystal, and then another difficult step on top of that will be to get the target proteins to dock into very ordered stereotype positions within each unit cell. So I think this is a fantastic goal for the field, maybe because it's very hard. I think it's important to have hard goals to reach for, but it may still take a while before we have a workable object that will actually help us with three-dimensional protein crystallization. In the meantime, what James Chu's group and my group have been able to do is to do something that's technologically much more modest, and yet it achieves the same end goal of enabling atomic resolution protein structure determination. And this is a method that's known as weak ordering. It's something I'll explain in a little bit, something that's been known about for a number of decades now, but hasn't really been well applicable to membrane proteins until recently we believe because of our tool. So host guest crystallization, you can think of that as a very strong sort of ordering, that we want to force the protein into very stereotyped translational position, very stereotyped rotational orientation. And that if that becomes too messed up, that's going to destroy the process of structure determination. In contrast, with weak ordering, what we're doing is we're just barely trying to change the population of solution tumbling molecules away from isotropic. So what does that mean? So in a nutshell, we have this animation to explain the concept. So imagine that the little red dots floating around represent our target protein of interest that we're trying to solve the structure of. And what we want to do is to introduce weak order into those proteins by mixing it into a dilute liquid crystal of these long molecular rods. We can zoom in here. The notion is that this is supposed to represent a membrane protein. This is the detergent micelle solubilizing the membrane protein. And the idea is that most proteins are not perfectly spherical in their shape, and they'll have a higher tendency to bump into these molecular trees if their long axis is perpendicular to the long axis of the molecular trees is perpendicular. And so therefore, if you could now mix your protein with a line sample of molecular trees, that should now provide a slight orientational bias so that the proteins tend to spend a little bit longer with their long axis pointing parallel to the aligning material than perpendicular. So in this case, usually you want these long trees to be about 1 to 2% weight to volume. And furthermore, for the NMR experiment, you want them to have a magnetic susceptibility so that if we put them in a magnetic field, then they'll basically globally align. And it turns out if you can now get your proteins to be partially aligned, you can now extract out information that otherwise would be invisible And that information can be very precise and allow you to calculate the atomic resolution structure of the protein. So I mentioned that this weak alignment method has been around for a while. Unfortunately, it's only been accessible to people studying soluble proteins because the most popular alignment media turns out to be a natural bacteriophage related to the M13 bacteriophage. And these work quite well for soluble proteins. We can make large amounts of them in bacteria. They have a magnetic susceptibility. They'll line up with each other. Works great. But the problem is that these bacteriophage will denature in the detergents that you need to solubilize membrane proteins. And therefore, we haven't been able to use the bacteriophage to weakly align membrane proteins because they just fall apart. That's where we came in. We decided we wanted to build DNA nanostructures that would be a shape mimetic of these filamentous bacteriophage. But because they're self-assembled from DNA, they should be impervious to denaturation by the detergents. And therefore, we should now make this method of weak alignment available to detergent-solubilized membrane proteins. On the upper left-hand panel, what we have are electron micrographs of our DNA nanotubes. In this case, we designed M13s to fold into a 6-helix DNA nanotube that's about 400 nanometers long, about 7 nanometers in diameter. We actually programmed 2 of them to come together to make a structure that is almost a micron in length. It turns out if you can make the structures longer, then they'll do a better job as these molecular trees. They'll actually line up more easily. And what we find is that when we concentrate them to about 2% weight to volume, then in fact they do start lining up. So this is an entropic phenomenon that's behind liquid crystal formation. And one signature of liquid crystal formation is birefringence. So if we look at our DNA nanotubes under cross polars, then we see this beautiful birefringence pattern, which is indicating that we're forming the liquid crystal. So when the sample is much more dilute, you don't see anything. But then now when the sample is concentrated, it forms the liquid crystalline phase, and then you can see this nice pattern. But most importantly, now if we take a test protein of known structure, so this is a transmembrane domain from the zeta transmembrane domain from the T cell receptor, we know what the structure is, and based on that known structure, what we can do is we can calculate what the kind of magnetic response will be for a partially aligned structure. So these are called dipole-dipole couplings. And then we can compare those predicted couplings against the ones that we experimentally measure when we now mix the protein with the dilute liquid crystal in the magnetic field. And then on the lower left-hand side, what we're doing is we're comparing on the y-axis the predicted couplings, and on the x-axis the observed couplings. And what we get is a very nice correlation between what we predict and what we observe with high signal-to-noise. We published this result in 2007. We were very excited about it because we knew that this meant that we had a tool that really works, that we could use this to solve the structure of membrane proteins. However, there's, of course, many different hurdles that still have to be overcome in order to solve the structure. But just to review, what we want to do is to have these molecular trees, or DNA nanotubes, that when we concentrate them to 2% weight to volume, they start lining up. We put them in an external magnetic field, so we get global lining up. We mix that with our protein of interest. The protein bounces off the rods, and we get that weak alignment. So we get that bias in the orientation of the population of molecules. Introducing that bias allows us to measure these dipole-dipole couplings that encode precise structural information about the protein, which we can then throw into a computer program that, if it has enough data, it can calculate the atomic resolution structure, at least of the backbone chain. Currently, we are not able to use this method to experimentally measure the configuration of the side chains. But even if you can just generate the atomic resolution backbone, then there are several very nice algorithms that allow you to predict how the side chains are going to pack onto that backbone. So Marcello Berardi in James Chu's lab was able to use our DNA nanotubes in order to solve the structure of a protein from the UCP family. So this is the uncoupler proteins that exist in the inner mitochondrial membrane, and they're known to have an activity of translocating protons back into the mitochondrion. UCP1 is the most famous member of this family. It's present in brown fat, and what it's doing is it's just leaking protons across that membrane, and that generates heat. So it's a mechanism to generate heat in brown fat. Marcelo solved the structure of UCP2. It's a related family member that's thought to be involved in energy source selection. So whether fatty acids versus amino acids versus pyruvate should be metabolized for energy as a source of energy. So he wanted to solve the structure. He tried for a long time using crystallography, using conventional NMR, but wasn't having a lot of luck. Of course, for any kind of structural biology problem, you first need to solve the problem of being able to overexpress your protein, being able to fold it to high homogeneity. That's always going to be difficult, and it took many years to do that. But to make a long story short, once he was able to generate a large amount of the protein in a homogeneous state, then he was able to mix it with our DNA nanotubes, weakly align the protein, use that in order to measure the dipole-dipole couplings, and then use that to calculate the atomic resolution backbone structure of the protein. And so this is here the crystal structure of this protein. It's a 6-transmembrane helix protein. Looking at the structure doesn't immediately tell you the mechanism of proton transport, but now James Shu's group is very interested in using this structure as a foundation for further structure-function exploration of what the mechanism might be. So the current hypothesis is that you actually have proton transported by these fatty acids that are flipping through the membrane. So when it's neutral, when it's protonated, it can flip through the membrane. But now when it's deprotonated on the other side, it can't flip back. So you're not actually going to get net proton transfer. So the idea is that the ionized fatty acid can now diffuse into the inside of this 6-helix barrel, and then you have a hydrophilic environment on the inside of that where the fatty acid can flip back. And therefore, this might provide a mechanism for multiple turnover of transfer of protons across the membrane by fatty acids. So that was a very satisfying experiment because we were able to demonstrate utility to a very urgent need in structural biology. And we're looking forward to further developments in the technology to make it more general, both for NMR experiments, also for cryo-EM, maybe X-ray crystallography as well. So the next area of applications that I'd like to describe are involving single molecule biophysics. So this is work making rigid handles for optical tweezing experiments that was done in the lab of Hendrik Dietz. He actually initiated this experiment when he was doing his postdoctoral training with my group at Harvard, and now finally was able to carry the project to fruition. It's going to be a great tool, and he was kind enough to include me on the author list. So the basic idea is, let's say that you want to use your optical traps in order to monitor the single molecule conformational changes of your molecule of interest. So let's say that it's a DNA hairpin that is opening and closing, and you want to be able to observe this. So ordinarily, this could be kind of hard to watch, but you also want to be able to watch this as a function of the force that you're applying on the ends of the hairpin. So you want to be able to dial in greater and greater amounts of force on my elbows to peel it away and watch what the effect is of that force on the binding and peeling and unpeeling kinetics of this DNA hairpin. So the way that you actually observe this is you now want to place your molecule of interest in between two large microspheres. And it turns out, for technical reasons, you can't have these microspheres too close together, so you need to give you some space. And in order to create that space, people like to use these double-stranded tethers that are on the order of 300 nanometers long. And so what you do is you have your beads, your tethers, and then your molecule of interest, and then you start to pull the beads apart. That generates a force on the molecule in the middle. And so if we now pull the beads further and further apart, that generates a greater and greater force on the molecule in the middle. And in principle, we can watch the opening and closing of that molecule in the middle by watching how the distance between the beads changes. So for example, we can imagine that if you have the molecule now opens up, now the beads should move further apart. If the molecule in the middle now snaps shut, then those two large beads should move closer together. And so by measuring the distance between the beads, in principle, we can infer the conformational state of the molecule in the middle. All right, sounds good. So where's the problem? The problem is that you have Brownian motion. So these large microspheres are actually undergoing a lot of motion, and it can be difficult to tell what is just random motion, what is reporting on something that's actually opening up in the middle. And the problem becomes especially bad when you have the very low force, because at low force, these double-stranded tethers are going to be very floppy, and you're going to get lots and lots of Brownian motion. So for example, on the lower right-hand trace, what we see is a time trace of the distance between the beads for the example on the top in B, where we're looking at a DNA hairpin that presumably is opening and closing at some specific force. And I'm not going to explain what's going on in the lower left-hand corner. I encourage you to check out the publication. But suffice it to say, if just looking at this bottom trace in E, you can't really tell when that hairpin is opening or closing. You can just see a lot of noise. In contrast, if we now replace those long tethers with a DNA origami bundle of helices, it's going to be much, much more rigid. So even at those lower forces, the Brownian noise is going to be suppressed. And so if we look at the same force at the opening and closing of this object in the middle, so D represents with these DNA origami handles, now we can hopefully tell that the noise is greatly suppressed, and we have some more confidence about assigning when the hairpin is opening and closing. So we think that this is a very nice application of these rigid DNA nanorod elements that will be useful for force spectroscopy, looking at single molecule dynamics and energetics of biomolecules. So the next area of application I like to call breadboard biochemistry, the notion that we can constrain the position of many different protein actors in a molecular play in order to understand and tease out their individual roles in the process. How are they interacting with each other? What are the stoichiometric requirements? What are the geometric requirements of this process? So the first example of this breadboard biochemistry I'd like to discuss is work that was led in the lab of Sam Peterson, my colleague at Harvard Medical School. It was done primarily by two students, Nate Dare and Brian Goodman. Nate is a student that was co-advised by myself and by Sam. And here we were interested in studying how ensembles of cytoskeletal or microtubule motors could antagonize each other in terms of determining direction of motion on a microtubule. And the motivation is that it's been observed that vesicles oftentimes bear both kinesins and dyneins, which of course move in opposite directions along a microtubule. And yet within a cell, one can often observe that the vesicles will choose one direction to go, and then remarkably will oftentimes switch directions. But what they don't typically do is just to stall. So how can we try to study this in a reduced system, an in vitro system? And as a first step, what we did was we self-assembled a chassis that is a 12-helix DNA nanotube that's about 200 nanometers in dimensions. And what we did was we decorated this chassis with single-stranded DNA handles that would come out at regular intervals at 7 different positions. And we can control and have any sequence we want come out at each one of these positions. In this particular case, what we did was we had 2 different sequences, one sequence for capturing dynein and another sequence for capturing kinesins. In this example, 2 dyneins and 5 kinesins. And the way that we capture them is that we express the protein, let's say a dynein, with a snap tag, and then the snap tag is used to capture an oligonucleotide with a snap tag ligand. And in that way, we're able to generate a protein DNA conjugate. And we purposely choose the sequence of that conjugate so that it will be complementary with the single-stranded DNA sequence that comes out of the DNA origami. So in this example, we're creating a chain gang of 2 dyneins and 5 kinesins, and we wanted to see what happens when we put this on a microtubule. Which way is it going to go? And what Nate and Brian found was that they basically stalled. There was an irreversible tug of war, at least in this first study. And furthermore, they're able to demonstrate if they introduced photocleavable elements either to release the dyneins dynamically or else to release the kinesins dynamically, then they could resolve this tug of war. So for example, in one experiment, if they release the dyneins, then now that stall would be relieved and the chassis would move towards the plus end of the microtubule. Likewise, if they caused the kinesins to be cleaved off, then now the complex would no longer stall and move towards the negative end of the microtubule. So we think this is a promising experimental platform for now further trying to find out what else do we need to add in order to recapitulate the very interesting behavior we see inside of a cell, where the vesicles, they don't just stall, but in fact they can move in one direction or another, and even more interestingly, change in direction. So here's another application of breadboard biochemistry where we're trying to study snare-dependent membrane fusion. So in this process, we have cells that are trying to fuse the vesicles with, let's say, the plasma membrane, and it's known that there are these transmembrane proteins that are mediating this called snare proteins. They have one domain that's, let's say this is the vesicle membrane, they have a transmembrane domain, and then a cytoplasmic domain, that's a coiled coil. And then in the target membrane, you have something analogous. You have a transmembrane domain and then a complementary coiled coil domain. There's two other helices that are involved as well. And so you can actually zipper up to form a 4-helix bundle, and that's thought to provide the energy for driving a vesicle in close proximity to the plasma membrane, because otherwise that's an energetically unfavorable process. And then once the two vesicle membranes are brought close together, then some other process happens that's not very well understood, causing the membranes to fuse. And there's some outstanding biophysical questions about this process that can be very simply articulated, but difficult to nail down very precisely. For example, how many different snare proteins does it take to trigger the fusion event? And then more subtly, how does the geometry of these proteins affect the kinetics of this process? And there have been some different measurements of the number of snares required. And depending on the context, it seems to vary between 1 and 10. In our case, what we're interested in is generating a more robust method for measuring these stoichiometric requirements. And therefore, we think we can use our system to study this problem at higher resolution. So here's the idea. Imagine you have a supported bilayer with your T-snares down there, and you're trying to now dock a vesicle on there. But you want to only have a controlled number of T-snares that could possibly participate in the reaction. So what if we were to create a molecular corral where we had 3 and only 3 of the T-snares in the corral? And now we could ask, well, is 3 snares enough for membrane fusion? So the idea here is that we chemically synthesize, we chemically link to the N-terminus of this snare protein, this white oligonucleotide in one test tube. And then in another test tube, we self-assemble this red DNA nanostructure. And the red DNA nanostructure, again, is decorated with these single-stranded DNA handles that are complementary to the white anti-handles. Now, when we mix the two together, we should get a controlled stoichiometry of the green whites onto the red, in this case 3. And now we can say, well, what happens when a vesicle tries to dock into the molecular corral? What happens when we have 3 snares in the corral? What happens when we have 2? What happens when we have 1? So certainly if we have no snares in the corral, then you wouldn't expect anything above background. And then as we start to increase the number of snares in the corral, hopefully the proteins now will be able to cooperate and to trigger the membrane fusion above background. And so I should mention that this is work that's still in progress. It's a collaboration between our lab and lab of Jim Rothman. Chenxiang Lin was a postdoctoral fellow in the group. He initiated the project along with Weiming Xu in Jim's lab. Now Chenxiang is an assistant professor at Yale, and in our lab, the project has been taken over by Bhavik Nathwani at the time of this taping. So again, what's thought to happen is that you have engagement of the T snares and the V snares, and then that brings the membranes close together. Some magic happens, membranes fuse. And that's the details of that biophysical process are very interesting, but we're starting off by asking a simpler question of just how many snares are involved and how does the geometry affect that. So just to prove our ability to decorate our DNA rings with different guests, we started with gold nanoparticles instead of snare proteins. They're just easier to see in the electron microscope. And we can demonstrate that we can now decorate our DNA rings with 3 gold particles on the inside, 3 on the outside, 6 on the inside, 4 on the inside, 8 on the inside. And what we've observed so far is that we can get something on the order of 90% occupancy of our sites. We'd like to get higher than that, something more like 99.99%. It's something we're working on. But currently, we think even with a 90% occupancy rate, this is still useful because it provides an upper bound on the number of snare proteins or whatever guess that could be bound in our assembly. So here what we're doing is we're actually binding snare proteins now, detergent solubilized snare proteins instead of gold nanoparticles. And again, what we're doing is we have these green snare proteins conjugated to a white oligonucleotide that are now self-assembling with this complementary red oligonucleotide being displayed on the outside of this DNA nanostructure. In our electron microscope, we can see enough contrast from the proteins to count the number of guest molecules on the DNA nanostructure. And then what we do is so in this case, what we're trying to do is create liposomes with controlled number of Snare proteins and see how they behave in a fusion assay. And so the next step here is that we mix our DNA rings with protein guests with giant liposomes in the presence of slight amounts of detergent. And through some process we don't quite understand, these DNA nanostructures with hydrophobic groups somehow take a bite out of the giant liposomes and end up with smaller liposomes basically filling the interior of the ring. So in this way, through a process we don't quite understand, we're able to capture liposomes on the inside of our DNA nanorings with controlled numbers of Snare proteins. And then here's a fusion assay that we use. It was developed by Erdin Kratikin at Yale. It's a microfluidic assay where we're fluorescently labeling these vesicles that we've captured in the DNA ring. And then when we get fusion, then what will hopefully happen is that those dyes will then diffuse out into the supported bilayer. So initially what happens is the dyes are quenching each other somewhat. So the first thing is actually the spot should grow brighter as the dyes dequench, but then as the dyes diffuse away from each other, then you should now get rapid dilution of the fluorescence response. And so we have here a microfluidic setup where we have an Eppendorf tube with our labeled vesicles that are being pulled through this microfluidic chamber. We're observing it using turf microscopy and then trying to see whether or not those vesicles can fuse to the supported bilayer as a function of the number of Snare proteins. So this is just an example of the assay in action. Your eye might be drawn to this giant blob on the bottom, but I'd like you to try to ignore that. Instead, focus on this red circle here, where you can see it's on a loop. So you can see a vesicle docking, and shortly after docking, then we get fusion. So that's the kind of event that we're trying to score. So far, we're still in the process of trying to see whether or not our scaffolded DNA rings actually can increase the rate of liposome docking and fusion. So hopefully we'll be able to make some progress on that for the next lecture. So the other class of applications I'd like to discuss with you involve potentially using DNA nanostructures as therapeutic delivery devices. And as a first step towards that, Franziska Groff, as a student co-advised by myself and Don Ingber, we set out to do some pilot studies to look at how the shape of DNA nanostructures might affect their uptake into a model cell line. In this case, these are umbilical vein endothelial cells. So how does the shape of DNA origami affect their tastiness to these cells? If you think about it, you could come up with a long list of potential descriptors that might make a difference. And it would require a very systematic study to look through all of these. But we just started out with a pilot study looking at aspect ratio. So in this case, we wanted to ask the question, if we have a bunch of particles of the same mass, but then we just made them in different shapes, then what would be the relative uptake? So let's first look at these three blue shapes that are highlighted in yellow. So they're the same mass, they're about 5 megadaltons, but they're made into a very long spindly rod that's a 6-helix bundle. And then we have these 24-helix bundle, and this is a 48-helix bundle that are successively more compact. So if we predict that structures with a longer aspect ratio should get into cells better, then we predict that this longer one should do the best in terms of getting into the cell, being taken up. In contrast, if we hypothesize that the more complex structures should get in better, then we'd expect to observe the opposite behavior, that this more compact structure should get in more quickly. And we also made some other shapes. So here we made a wireframe octahedron. We also made 3 different orange shapes that are analogs, but now just 40% the mass. And so we could say, well, with this starting panel of 8 structures, what's the relative rate of uptake into these HUVEC cells? And what we observed, so I have a very busy slide here, but I'll try to give you the overview. So first thing is, if you just kind of squint your eyes, you might notice that the bars, which represent some kind of uptake, tend to be taller on the right-hand side of the slide than on the left-hand side of the slide. And also, if you look at it for a little bit longer, you might notice that the structures that are on the right-hand side tend to be more compact. than the structures on the left-hand side. So just from simple eyeballing of the figure, we can get the feeling that, at least for this class of particles, when the structures are more compact, then they get in more easily into cells. So a second order piece of information that we learned is, so you might notice here that there's hollow bars and solid bars. So what does that mean? So the hollow bar represents the amount of fluorescently labeled nanostructures that were taken up by the cell after some incubation time, 16 hours. And then the solid bar represents the same experiment, but what we did was we treated the cells with DNase-1 before we did the fluorescence analysis. So what that's going to do is to remove any membrane-bound DNA nanostructures, and now the assay will only report those structures that actually have been internalized, that are now protected from DNase digestion. And so what we observe is something quite interesting, that for these compact structures, the height of the bars is very similar. So that says that most of the particles that stick to the cells immediately go in, or getting in quite efficiently. But for these extended structures, you might notice that the hollow bar is actually much taller than the solid bar. And what that suggests is that if you're really extended, now maybe you can resist getting internalized, which we can rationalize by saying for this class of cells, they're going to have they don't really have good mechanisms for engulfing structures that are 400 nanometers long. So this might present an interesting opportunity therapeutically, that we could design structures that have the tendency to persist on the outside of the cell that can resist internalization. It might be useful for creating a sentinel or a beacon for other nanoparticles to then deliver their contents to that particular cell, because if you just get swallowed, then you're not going to be able to act as that sentinel. Here what Francisca has done is she's taken one of the structures, this nanocylinder, and she's decorated the outside of the nanocylinder with a bunch of ligands, in this case cyclic RGD ligands that are known to bind the alpha V beta 3 integrin receptors that are overexpressed on the cell line. So the prediction is that if we decorate our nanoparticles with a high density of these ligands, then we're going to increase the rate of internalization. And we can also do a control with cyclic RAD peptides that should not interact with those receptors. And in fact, that's basically what we observe, that when we have the cyclic RGD labeled structures, we get an order of magnitude greater uptake of these particles compared to controls that have no peptide or controls that have the mock peptide sequence, cyclic RAD. And so what this says is that we have the ability to make these particles of different shapes to either help their uptake or help prevent their uptake. We can decorate these particles with a high density, controlled density of ligands to help further modulate that process, either get faster uptake or not. So far I've shown you naked DNA particles. Again, we might be worried about things like nuclease digestion of these particles. We might have a desire to encapsulate soluble factors in these structures. So if we try to make a wireframe cage and then we put soluble factors on the inside of the cage, then those soluble factors might just diffuse out through those windows. So how do we keep those factors on the inside? So Steve Corral in the group has pioneered a method for encapsulating these DNA nanostructures within liposomes to create something that's structurally analogous to an enveloped virus. So the way that he's done this is he self-assembles a DNA octahedron with single-stranded DNA handles coming out, and then he hybridizes on a complementary oligonucleotide anti-handle that has a lipid conjugate covalently linked to it, solubilized by detergent. And so through base pairing interaction, he basically gets these detergent solubilized lipids to cover his DNA octahedron. He has something like on the order of 50 copies of these lipids covering his octahedron, solubilized by detergent. The next step is that he mixes this, he dilutes this into a solution of giant liposomes, and then dialyzes out the remaining detergent. Through a process that we still don't quite understand, we get shrink wrapping of the liposomes around our DNA nanostructures, creating something, again, that resembles, under the transmission electron microscope, envelope viruses. So here we can see on the left the naked DNA octahedra. They're about 50 nanometers in diameter. And then on the right, we can see the liposome encapsulated DNA octahedra. Again, very reminiscent of envelope virus. So we think this is an important step towards the versatile use of DNA nanostructures for delivery for example, of soluble factors, or if we wanted to simply protect the DNA from nucleases or factors that would try to digest it. We, of course, still want to be able to decorate the surface of the structures with functionalities. So now we're working on the ability to present transmembrane features starting from the inside, going through the membrane, and then basically controlling the spatial orientation of ligands through the puppet master on the inside. And then the final thing that I'd like to show you is actually not from my laboratory. It's from Sean Douglas when he was a postdoctoral fellow in George Church's group. And he did this along with Ido Bachelet, they did this fascinating pilot study where they generated something they called a DNA origami nanorobot that was designed, at least in this test tube example, to specifically recognize cancerous lymphocytes and kill those off, and program them to commit suicide while leaving the healthy cells alone. So how is this supposed to work? The idea is that Sean designed this DNA barrel that's about 60 nanometers in diameter, and he placed on the inside antibodies that are known to bind to receptors and cause receptor clustering that then leads to apoptosis. So kind of like what a natural killer cell might do. But in this case, because the antibodies are sequestered on the inside of the barrel, they're not actually accessible to the cells. The cells have fat fingers, if you will, so they can't actually reach in and touch those antibodies. And the notion is that Sean and Ido wanted to program this robot to open up when it encountered the cancerous cell, but not when it encountered the healthy cell. And if the robot were to open up, then now the antibodies that trigger apoptosis could now be accessible to the cell surface. So how do they get this robot to open up only in the presence of the cancerous cell, but not in the healthy cell? So what they did was they designed this lock and key mechanism where they had single-stranded DNA on the two ends that would hybridize together. And they designed the sequence with something called a structure-specific aptamer. And what happens is that this aptamer recognizes some protein ligand. And so basically that protein ligand is competing for interaction between the partner double helix, partner strand. So in other words, these two strands will interact with each other, but that can be competed off by some specific protein ligand. And they used sequence on one of the locks that was derived from the literature, a sequence that somebody else had isolated through a SELEX experiment. That sequence was known to bind to EGFR, which is overexpressed on their sample cancer cell line. And then they found in the literature another sequence that was found to recognize something that's enriched on their cancer cell line, but not on the healthy cell line. So they're able to do a logical AND statement here. Only if the target cell could open both locks would the shell open up and then reveal the antibodies to the cell surface. So in this way, you could argue that it's an intelligent robot and that it can do this more complicated logical argument. And they're able to show, at least in the test tube, that in fact this nanorobot seems to trigger the apoptosis a couple orders of magnitude more easily than the healthy cell. Of course, moving this into an actual therapeutic environment requires several hurdles, because if you wanted these things circulating in your blood, you'd want them to avoid nucleus digestion. You'd want them to avoid clearance by the immune system. So of course, there are going to be many hurdles to overcome in order to translate this to the clinic, but we think this is a fascinating first step in that direction. So to conclude, DNA nanotechnology is more than just smiley faces, we think. In particular, we're very interested in two classes of applications. One is as tools for molecular biophysics, whether it be in tools for structural biology or especially single molecule biophysics. And secondly, we are exploring the notion that these DNA nanostructures might be useful as therapeutic delivery devices. And of course, there's many different platforms that scientists are trying to explore for delivery, but we're motivated by the insight that our immune systems, in fact, can be thought of as very complicated nanotechnologies. They can process lots of information. They can actuate all kinds of things. They can punch holes into cells. They can program each other to expand. They can squeeze through tight spaces. And the only way to achieve this really diverse, sophisticated behavior is by creating complex nanoscale objects. And at the moment, we believe that the DNA nanotechnology platform provides a very powerful method in that direction. So I'd like to thank the following sources for support, especially NIH and Office of Naval Research. I also got some support from the Wyss Institute for Biologically Inspired Engineering. I've tried to acknowledge the different folks who've been doing the work on the slides describing the work. Thanks a lot.